Right. Hi, everybody. First of all, Aaron was really sorry he couldn't make it today to introduce Elan. But I'm Sterling. I'm an RA in the plant lab. I've also had the pleasure of working with Elan over the last 10 months or so. And I'll help narrate the questions at the end as well. Without further ado, I'll give the introduction. So I'm pleased to introduce today's speaker, Elan Tran. So Long grew up in Delaware and graduated from the University of Miami in 2021 with a bachelor's degree in microbiology and immunology. In September of last year, Elan started her internship as an NSF REPS fellow, co-advised by doctors Michelle Afkami and Chris Searcy at the University of Miami. With her interest in plant pathology, we tasked her with being the fungi person in the lab, and since arriving, Elan has been nothing short of amazing. She was first tasked with spearheading our new macrofungi collections, or the fungarium, and she rose to the challenge, establishing lab collecting and processing protocols. Soon after, the colorful Exobasidium galls that appear on our Lyonias each fall caught her attention, and she focused her intern project on them, which we'll discuss here today. And finally, in her last few months here, she designed and implemented our first study of the mutualistic ectomycorrhizal fungi that associate with many of the shrub species here. In addition to being a wonderful member of the plant ecology program, she's been an important presence at the station. She served on the DEI committee, assisted with several camps, outreach events, virtual field trips, and written a newspaper article. Next month, Elan will begin her PhD in plant pathology at the University of California, Berkeley, which will be funded in part by a very prestigious NSF Graduate Research Fellowship. So after a wonderful 10 months here at Archbold, we're excited to see what amazing things Elan will do next. With that, I'll turn things over to Elan.
That was very sweet. Thank you, Sterling, and thank you, Aaron. All right, I will just get started. 1 second. Okay, so hi, everybody. I'm Elan. Today, I will be presenting my intern project titled How Fire Feeds Fungi, Fire-Dependent Infection by Exobasidium ferrugineae on Lyonia fruticosa in the Florida scrub. And so before I get into the details of my project, I'm just going to give a brief overview about fungi and a brief overview about fungi here at Archbold. And so fungi belong to their own kingdom of life. They're actually more closely related to the animal kingdom than they are to the plant kingdom, which is interesting. And there's many types of fungi. There's yeasts and molds and rusts. So only some of them are actually mushroom producing. And fungi are incredibly diverse. They have diverse genomes, diverse morphologies, and diverse life cycles. So building off of that point, this diversity allows them to contribute to our ecosystems in different ways. So just in their interactions with other organisms, they might be commensals, mutualists, or pathogens. And so one of the lab projects that I've been working on is to look at the fungal diversity here at Archbold with a main purpose to document the diversity of mushroom-producing fungi on the Lake Wales Ridge. And so this has been a really wonderful project to be a part of. You can just see the diversity of just some of my specimens that I've collected. I've collected, poisonous species, edible species, species that produce liquid latex. It's just been overall a really great time. And so in addition to looking at the diversity here at Archbold, I've also helped on a lab project to look at the fungal mutualists here. Specifically, we're looking at these fungal mutualists that form these relationships with plant fine roots underground. And we're looking at this relationship at 5 oak species and 1 hickory species. So that's just getting started, and I'm really excited to see where it goes. Okay, so I've talked about the diversity, I've talked about mutualists, but what about fungal pathogens? And so this is what I did my intern project on. If you have been at Archbold in the fall, these images might seem familiar to you. In the fall, we get these really large, colorful structures on Lyonia fruticosa. And I was really interested in this. And it turns out that this, physical description closely matches infection by the fungal genus Exobasidium. And so I set out with a couple objectives to investigate that further. But first, I'll just let you guys know a little bit about Exobasidium. So Exobasidium is a fungal genus that contains many pathogenic species that often infect and form galls on plants in the order Ericales. And so a lot of the research that's been done on this genus so far is focused on crop plants, specifically blueberry, cranberry, and tea. So on the right, you can see pictures of some blueberry that's been infected, specifically the flower and the leaf. And so in addition to crop plants though, Exobasidium can also infect native shrub species. And there are some similarities between these infections. And so infection is usually marked by hypertrophy of the plant tissue. So this just means that the cells grow really large. And that's why, the small leaf has become this giant gall. And then the infection is also usually marked by a change in color. And so life cycles of these species are very complex and they make our knowledge often incomplete. Okay, so my three objectives were to molecularly identify the pathogen, determine factors driving infection, and to determine effects of infection on host growth. Okay, so let's start with the first, molecularly identifying the pathogen. So the first step in this is to actually isolate the fungal tissue. So I did this from a couple different galls on different Lyonia fruticosa plants. And then I extracted the fungal DNA from that tissue and amplified it with PCR. And then once I had that amplified DNA, I sent it off for sequencing of two specific gene regions. They're called the large subunit and the internal transcribed spacer region. And so then once I got those sequences back, I could compare them to other fungal sequences in a database. And before I move on to show what my sequences came up with, just wanted to mention a little bit about this process. It looks very straightforward in this diagram, but wasn't in real life. Just isolating the fungal tissue was kind of difficult. Exobasidium has known to be really hard to culture, so I tried that and that didn't work. And then I just tried taking chunks of the gall, but I think there's just like too much plant material, so that didn't work either. And eventually I tried just scraping the top of the gall, and that's what worked the best. And I think it's because if you see this kind of like whitish sheen on the leaf, that's all of the spores that the fungus is releasing. And so I think just by scraping the top, I was able to get the highest concentration of like pure fungal tissue. Okay, so what did the sequences show? So here is a phylogeny that I constructed from the large subunit region that I sequenced. And you can see that my samples are a match for Exobasidium ferrugineae. And if the last part of that sounds familiar, it's because Exobasidium ferrugineae is known to infect Lyonia ferruginea. And so this is the only host that's been documented for that specific pathogen so far, which means that since I also sequenced it, but on Lyonia fruticosa, that I have identified a new plant host for this pathogen. And so that was really exciting. And this, this affects the epidemiology of this pathogen. So you have to think about if there's two hosts, it can spread in new areas. There's now two host reservoirs. So that was really awesome. Okay, so now I knew what the pathogen was. I wanted to know what was driving infection. And so I decided to look at a couple of factors. The first was time since fire. I looked at this over a broad fire gradient from less than a year to 10 years. And originally I was thinking that as time went on, that fungal infection severity would increase because there would just be maybe a buildup of inoculum or something like that. I also looked at host density. So, maybe the more amount of Lyonia fruticosa there is and the closer it's together, that it's easier for the pathogen to hop from one plant to another. I then looked at elevation. Maybe areas of lower elevation are more moist and are benefiting the pathogen, because of its moist environment. And the last is distance from the edge of the fire. So I thought that maybe the edges of burn units would have a higher incidence because of trucks going by and just maybe more foot traffic, that there would be more chances of inoculum spreading to these areas. And so to test this, I set up a field survey in 12 management units or burn units. And at each burn unit, I surveyed 6 quadrats that were set up in 20-meter increments from the road. So at 0 meters, 20, 40, 60, 80, and 100. And then the last thing to note about the survey is that host density was measured in number of trunks per quadrat. Okay, so the main result from this part of my project is that time since fire is the strongest predictor of infection severity. So I ran different models, and it turns out that the best model, or the model that best fits my data, solely uses time since fire. And so what that means is that those other factors that I mentioned on the previous slide, that when you try to model or create models with those factors, including them doesn't help create a better fit. And so that's how I know that time since fire is the strongest predictor of infection severity. And infection severity was measured in galls per infected trunk. And so something that's interesting about this image is that you can see that infection severity is higher in the first couple years after fire. And I think this has to relate to some published information on other Exobasidium species. And some published studies have shown that Exobasidium has a preference for infecting young and tender plant tissue. So after fire, there's going to be an abundance of new growth and young and tender plant tissue. So I think that this could create a strong relationship between time since fire and infection. Okay, so now we know what the pathogen is, we know a little bit about what's driving its infection. Now I wanted to know, what effects does it actually have on Lyonia fruticosa? Is it actually detrimental to this plant? And if so, how much? So I created a field survey where I assessed 3 burn units that were less than a year to 2 years since fire. And I looked at flowering and shoot growth from January to March of this year. And so for this field survey, I looked at 3 different stem types. I looked at on infected plants, infected stems on that plant, and uninfected stems, so those were paired. And then on uninfected plants, I just looked at their uninfected stem as the control. So we have uninfected stems on infected plants, infected stems on infected plants, and uninfected stems on uninfected plants. Okay, so my first result from this is that infection is associated with lower flowering. So I ran analysis for January and March, and these were run separately. And so in January, I found that the infected and uninfected stems on infected plants had significantly lower flowering than just the control, the uninfected stems on infected plants. And then in March, the infected stems on infected plants had significantly lower flowering than the uninfected stem on uninfected plants. And so in addition to a lower presence of flowering, there's also a lower flower count. So actual number of flowers and number of flower buds. So this shows that in March, the uninfected stems on infected plants had significantly lower flower counts than the control of the uninfected stem on uninfected plants. And so, oh, by the way, this is of stems that were flowering. That's what this analysis is based off of. And this is kind of interesting. I wasn't exactly expecting this result, but it suggests that infection causes some type of systemic effect on the host that's causing the, or that relates to an uninfected stem on the infected plant to have a lower flower count. So that was pretty interesting. And so the last thing I wanted to touch on has to do with the apical meristem infection. So the apical meristem is at the shoot apex of a shoot, and it's just that area where there's new growth and cells are dividing. And so when I was in the field, these infections were pretty fun to look at because they just turn into like these really crazy, I mean, this is a dried out one, so it's, you know, dark, but when they're red, they're usually really large and have like all of these different gall blobs on them. But unfortunately for the plant, a lot of the times when they're infected in this area, they don't recover. The shoot just never grows, opposed to just having a leaf infected and maybe that like leaf falls off or something like that, but it doesn't really have other repercussions for that shoot. And so in this graph, I'm just showing the probability of a infection in this area have like the probability of a shoot with this type of infection growing afterwards. And so you can see in January that this was quite low, and even in March it was still quite low. And so even though I don't have any control against this, I still thought it was quite striking that this shoot that was supposed to grow has a very low probability of growing again. Okay, so my main conclusions are that Lyonia fruticosa is a newly documented host of Exobasidium ferrugineae. Exobasidium ferrugineae infection is negatively associated with time since fire. Infection was associated with decreased floral production and low shoot growth. And these can all be wrapped up in a major takeaway that this study reveals the fire dependency of Exobasidium on a native shrub and highlights the interplay between fire, plant recovery, and fungal pathogen infection. Okay, so I just want to give a bunch of acknowledgements right now. Of course, a ton to Aaron for just guiding me throughout this whole entire process. He was a really great mentor, as well as everyone else in Plant Lab. I'd also like to thank my co-advisors, Michelle Cami and Christopher Searcy at the University of Miami. I'd like to thank Matthew Smith and Laurel Kaminsky at the University of Florida. They were such a big help with setting up the workflow for the fungal herbarium. They helped me with sequencing for the fungal herbarium and sequencing for my Exobasidium project. I'd like to thank Vivian and Julie for help with GIS, Nova and Sharon for everything that they do, all the interns for just being amazing people, the National Science Foundation for funding and support. And lastly, just a special thank you to the DEI working group. This has really made a difference in my time here at Archbold. I really looked forward to getting on those Zoom calls every week, and this type of DEI work group is really something that I want to be part of no matter what organization I join next. And so thank you all for listening. And with that, I will take any questions.
All right, thank you, Elan. Very cool talk. I can read some of these questions in the Q&A.
Yeah.
All right, so the first question I got was from Josh Daskin. So he says, interesting resusceptibility of plants in recent post-fire areas. Could one do an experimental transplant to confirm plant age as the main mechanism? And he asks a second question. Are there other pathogens of plants or otherwise that are more prevalent after fire at Archbold?
Okay, so the first question, talking about an experimental transplant, that would be really interesting. And yeah, I would love to be able, with more time, to do something like that for this pathogen. Like I said, a lot of Exobasidium, there's a lot of question marks when it comes to certain parts about their life cycle and things like that. So yeah, that would be really interesting. And then, are there other pathogens that are more prevalent after fire at Archbold? I'm not sure about at Archbold. I was reading some other papers about other post-fire pathogens, and it can really go either way because fire can sterilize soil, but then there's also some studies showing that after fire, there are other fungal pathogens that take over. So I don't think, this is the first pathogen to do this. but it's definitely an interesting result, and it would be really cool to see if there's other pathogens here that follow the same trend. Oh, you're muted, Sterling.
Whoops. Next question is from Carol. How does the pathogen spread its spores? Is it through wind?
So kind of like I mentioned in the previous question, a lot of its life cycle, even transmission, isn't really known. There have been studies that have found aerial spores, so these could be, you know, transmitted through the wind, but they don't think this is actually the primary inoculum causing infection. And there is one idea which I just really like the sound of, and it's that the infection is possibly spread through pollinators. So when I was first showing those images of Exobasidium, these really like large red, like probably to a pollinator attractive looking structure, um, maybe some type of mechanism that helps to, I don't know, get certain insects attracted to the structure and then they go off and spread it. So that would be really cool. Yeah.
Cool. Next question is from Akeem. Did you expect lower flowering on plants with infected stems because other fungus cause this on other plants? Also, did leaf count or any other leaf traits change with infection?
So for the last one, I did not measure any other leaf traits. That would be interesting though. But yeah, I didn't specifically look into that one. And then did I expect lower flowering? The main reason why I expected or thought that there might be an effect on flowering is because of those, apical meristems. That whole shoot was supposed to possibly grow and then flower. And if that whole shoot, dies, that's going to have like a pretty big effect on the reproductivity of the plant. Um, does it cause lower flowering count? I haven't seen like a specific study linking like the exact flower count, but I know that on the crop studies, it destroys the fruit. So that's like another problem.
Cool. Okay. Next question is, I guess, Skylar on behalf of Christine. Why do you think the infection happens in the fall?
Yeah, that's really interesting. So I don't know for sure, but I think one thing that can contribute to it is that other Exobasidium spores can't survive temperatures that are higher than like 80 degrees, or I think it's like 82 or something like that. And so that might be why they kind of over summer when it's really hot, and then maybe in the fall when the temperature starts to drop is just when they're like able to survive and actually produce infection.
Maybe that sort of answers the next question from Shri. Why did you choose January and March for your sampling?
Yeah. So the actual like field survey of infection was done in the fall, and then January and March is just because that's when the plants were flowering, so I could measure that.
Cool. And then a question from Rosa. What is your process for preserving mushrooms in the herbarium?
Oh, the herbarium. It's pretty straightforward. So when I get a specimen, um, I usually put it in the dehydrator for like 24 hours. You just really want to get all the moisture out. And then you'll want to freeze it for at least a day to kill like any insects that could cause damage later. And then you just store it in a dry place with some desiccant and it is good to go.
Nice. Okay, we have a question from Andy. It looked like there were some plants with infection in management areas that had burned less recently. Would you expect differences in flowering/stem growth based on time since fire?
One second, I'm just rereading it.
Yeah.
Yes, I think so. I don't know a ton specific just about like Lyonia fruticosa's entire life cycle. And so I think that definitely could be the case where, maybe there's a like a higher rate of growth right after fire, and then maybe it levels off after a couple of years or something like that. That would also be something really interesting and important to take into account with this.
Definitely. Next question is from Haley Dole. She says, great job. Do all infected plants form galls?
I believe so. I haven't seen, any other indicators on the plant that they are otherwise infected. It really just seems to be these like concentrated gall infections. In other species, they'll sometimes have different characteristics, like they might just form a spot on the leaf and not necessarily a gall. But with Lyonia fruticosa in particular, I think it's just galls.
Interesting. There's a question from Aiden. Does this fungus benefit the host at all?
Not to my knowledge. It would be interesting if there is some type of like cost benefit relationship. Like, this is all speculative, but maybe there's a cost to the host because it maybe like lowers flowering or decreases shoot growth, but then maybe there is some type of like benefit. So it's trying to like, navigate those two. But I don't know.
Cool. Any other questions for Elan?
All right, then.
No.
With no more questions, I guess we'll close. Great job, Elan. Thank you so much. Thanks to everyone for attending today. So join us again on Thursday for another intern seminar presented by Jack Christie entitled Movement and Behavior of Gopher Tortoises During Nesting Season. And a week from today, next Monday, tune in again for an avian ecology intern double feature by Skylar and Brian. The link to that is coming soon. It'll be on our website, hopefully by tomorrow. So anyone who wants to sign up for that, please check our website or email me, Laura, at execassistant@archibald-station.org. Thanks everyone again. See you soon.
Great job.